Hi, my name is Gloria Roberts. Today we are looking at the topic microorganisms around us. Objectives of lesson. By the end of the lesson, students should be able to 1. Define the term microorganism. 2. Mention the types or groups of microorganisms. 3. List the types of bacteria based on shape. 4. Define culturing. 5. Define aseptic techniques. And 6. State the procedures involved in culturing. What are microorganisms? The word micro stands for small or tiny, while organisms are living things. So microorganisms are very tiny organisms that cannot be seen with the naked eyes, but by the use of an instrument called microscope. Here is an image of a laboratorist observing microorganisms through an instrument called microscope. Microorganisms can also be called microbes, or commonly we call them germs. Now, microorganisms exist almost everywhere, such as in water, in the air, in the soil, surface of objects such as on clothes, on rails, metals, and so on, as well as on and inside the bodies of organisms. So, microbes can be found in the ear, in the mouth, in the nose, in the eyes, on a palm, on the skin, and even inside the body. They occur most abundantly where there is food, moisture, and adequate temperature for their growth. So when you have food, water, and warmth, microorganism will definitely be in such a place. Types of microorganisms. Different types of microorganisms are 1. Viruses. 2. Bacteria. 3. Fungi. 4. Algae. And 5. Protozoa. Now we're going to be discussing the types of microorganisms one after the other, and we are starting by looking at bacteria. Bacteria usually exist in clusters or colonies. Each bacterium cell has a slimy capsule. It has cell wall with cytoplasm, but they lack true nucleus. Now the nuclear materials are DNA. So here is the structure of how a bacterium look like. Now take your time and study the structure, how to draw and how to label well. So let's discuss types of bacteria. Bacteria are grouped into two categories. The first category is categories based on use of oxygen. Number one, we have aerobic bacteria. These are bacteria that make use of oxygen for their respiration process. 2. Anaerobic bacteria. These are bacteria that do not require oxygen for their respiration process. And 3. Facultative bacteria. These are bacteria that can carry out respiration process with or without the presence of oxygen. So the second category based on shape. Now we have different types here. The first one we have the cocci. Now singularly we call them coccus. Now these are bacteria with spherical shape, or they can be said to be round in shape. There are several forms of this kind of bacteria. The first one we have the streptococci. Now if you have one, you call it streptococcus. Now these are arranged in straight chains. When you have several coccal bacteria come together and they form a straight chain, what they form is what we call streptococci. Now they are sore throat causing bacteria. The second one here we have the staphylococcus, okay, or staphylococci if there are many. Now these are bacteria that stick together to form irregular bunches or clusters. They form a kind of ball-like structure. Now they are boil-causing bacteria. We also have the diplococci. These are called in pairs. E.g., we have the pneumococci, which causes pneumonia. If you have only one bacteria, you call it bacterium. If you have many, you call it bacteria. So take your time and study the different kinds or forms of the cocci or round shape bacteria. Now, other forms of bacteria. Number 2, we have bacilli. Singular, we call it bacillus. These are rod-shaped bacteria. They look like a small rod. They cause typhoid fever. Now, if you look at the second row, the last one there is the bacillus. So, several, you call it bacilli. One, you call it bacillus. Number 3, we have the spirilla. They are spherical in shape. So singular, you call them spirillum. All right. The first image on the last row is an example of spirilla. Number 4, we have the vibroids. Okay, these are comma-shaped bacteria, and a common example is the Vibrio cholerae, which causes cholera. Number 5, we have the Spirochaetae. These are helically coiled bacteria. They are sort of like spiral bacteria, but they have more coils than the spiral shaped bacteria. Example is the Treponema pallidum, which causes syphilis. Treponema pallidum, which is responsible for syphilis. So the other types of microorganisms, we also have protozoa. Number 2, we are looking at protozoa. Protozoas are common in freshwater and moist soil. Examples include Euglena, amoeba, paramecium, trypanosoma, and plasmodium. These are the common examples of protozoa. Here's an image showing different categories or shapes of protozoa. The third microorganism we are looking at is fungi. Now, fungi lack chlorophyll, so that means they cannot carry out photosynthesis. Thus, they are usually called saprophytes. Now, the meaning is that they are responsible for causing the decay of dead plants and animals. Now, because they feed on them, they can grow on oranges, lemon, cheese, and even bread when they are getting spoiled. Now, examples of fungi include mucor, mushroom, rhizopods, penicillium, aspergillus, toadstool, rust, and so on. Okay. Number 4, we have viruses. These are another categories of microorganisms. In short, they are the smallest among all set of microorganisms. Now, viruses are so small that they can only be seen with electron microscopes. They are enclosed within a protein coat and consist of nuclear materials such as DNA or RNA. Outside living organisms, they are like non-living things, right? But when they get into the body of a living things, they begin to carry out characteristics of living things. Now, examples are the coronavirus, which is trending now in our environment. We have the HIV virus. We have the influenza virus, hepatitis B virus, Ebola virus, the Toga virus, and so on. Now, number 5, algae is another form of microorganism. Algae have chlorophyll, so that means they are green in color. They can carry out photosynthesis. They can manufacture their food by themselves. They occur abundantly in water, moist soil, barks of trees, stones, etc. Okay. Now, free floating microscopic algae are referred to as phytoplankton, and they form the major food of aquatic animals. Now, examples of unicellular algae include chlamydomonas, protococcus, and so on. Ways or places through which microorganisms enter the body. Microorganisms can enter the body through different routes or channels, such as number 1, natural openings. Certain natural openings, such as anus, the ear, the nose opening, the mouth, are channels through which microorganisms can enter the body. Now, number 2, animal bites. When some animal bites, they can transmit microorganisms from one person, from themselves, or to another person. Then, 3, blood contact. When blood of an infected person gets in contact with someone that is not infected, such a person can become can get infected by the microorganisms in them. Then number 4, genitals. That is through sexual intercourse. Okay, the genital organs. Okay, the fluid in the genitals can help to spread microorganisms from one organism to another. Now 5, birth canal and the umbilical cord. This is from mother to unborn child and so on. Then number 6, body contact. Okay, through body contact. Right, now here are images showing male and female and the channels or routes through which microorganisms can enter the body. So take your time and study them. Okay, let's discuss concept of culturing. What do we mean by culturing? This is the art of growing microorganisms in the laboratory in a special medium. So as a laboratorist, if they want to grow microorganism in the laboratory, they have a method of doing that, and that method is what we call culturing. All they need to do is prepare a special medium and put the microorganism in. After a while, there'll be growth of this microorganism, which they can now use to carry out their findings or experiments. So culturing actually involves the techniques of preparing the medium. That is the first stage. After preparing the medium, okay, it must be the microorganism must be inoculated. So that means involved inoculating, transferring of the microorganisms into the medium. Then the third one is incubating, ensuring that the right temperature, the right environmental conditions are available for the microorganism to grow. And the last stage is examining the microorganism, their characteristics, their appearance, and the rest when they are being grown. Okay, the material in which microbes grow in the laboratory are called culture medium. Culture medium can be prepared by using two components. The first one is alga. Alga is a jelly-like structure, and the second one is by using extract of meat, like meat stock, fish stock, and the rest, vegetable, and that of yeast. All right. Here are images, two different images. The first one is showing a prepared medium, okay, containing maybe alga, extract of beef or vegetable, and yeast. And then the second one is showing the end if the microorganisms are being inoculated into the medium, their growth, okay, after a while is showing the growth of microorganism. Okay, we have types of culture in the laboratory. We have the pure culture. This is a culture that contains only one species of organisms regardless of the number of individuals. For example, if you want to culture bacteria, okay, and in that bacteria you have just bacteria, no other forms of microorganism, you know that such is a what? A pure culture. Now mixed culture, this is a culture which contains two or more species of a microorganism growing together inside the petri dish. So now note that cultured microorganisms, they grow in colony, and each colony can be recognized by their colors, pattern of growth, appearance, and so on. So let's talk about aseptic techniques. This involves the various procedures that are taken to ensure that the species of microorganism that is being cultured, okay, they are not contaminated by any other microorganisms from the surrounding, such as from the air, the water, hand, cloth, apparatus, and materials that are being used. All right. Now, the aseptic techniques include sterilizing culture media and all glassware that will be used. Okay. Now, when culturing is to be done, the materials, the glassware to be used, they must all be sterilized. Number two is that keep all petri dishes, flasks, and test tube that are involved covered to ensure that they are not contaminated. Three is to open petri dishes, flasks, and test tubes near presence of a blue Bonzi flame for a short period of time to make sure that any external or not unwanted microorganisms are destroyed before culturing. The number four is flaming all equipment used for the transfer of microorganisms just before they are used. Okay, now this ensured that the microorganisms that are not necessary or needed or that can cause contamination are destroyed. Number five is to wipe the work area with cloth or cotton wool soaked in a strong disinfectant before and after use. Then number six is to avoid breathing on culture or sterilized instrument. Of course, this is by the laboratories. The scientists involved must ensure that it doesn't breathe into the because there are airborne microorganisms in the nose or mouth. Then the last point here, which is number six, is to perform experiments as rapidly as possible. This is to make sure that contamination is minimized. Sometimes the environment might not be conducive, might not be able to actually totally eradicating contamination. But if experiments are done rapidly, at least it can minimize contamination. All right, let's look at some equipment used during culturing of microorganisms. The first one here is the petri dish. This is where the medium is prepared and the microorganisms are placed in. We have the inoculating loop. That is a loop that is used for picking the microorganism and putting it inside the petri dish. We have a face shield. This is to prevent the laboratories from breathing in microorganisms easily that can cause infection, and also from ensuring that the scientist does not breathe into the cultured material. So it's a form of protection. Then we have flags for collecting materials. We have microscope, very important instrument which is used to see these organisms because they are very small. We have the incubator. Now, incubator is a machine that is used to ensure that the microorganisms that are being cultured are exposed to the right environmental condition to ensure their growth. We have hand gloves. These are another protective devices, or let me say equipment that ensures that the hands of the scientist is protected. We have test tubes and of course the test tube holder. We have other equipment such as the slide cover, which is used to observe the microorganism through which is used to keep the microorganisms before they are observed through the microscope. All right, you can study more equipment, okay, as many as you get to have a better understanding. All right, we have carriers of microorganisms. Now, any agent that carries microorganisms from one place to another, this agent or that agent is called a carrier. Now, carriers can be living or non living things. So, when you have non living carriers such as air, water, and food, okay, now we call them the non-living agent or the non-living carrier. Why animals are also carriers, but they are called the living carriers. So these animals or the living carriers are what we call the vectors. Okay. Now vectors are animals that carry and can spread diseases causing microorganisms from one animal to another. Vectors are carriers of disease causing microorganisms from one organism to another. Now note, I didn't define vector as disease causing microorganisms. The vector themselves are not responsible for causing diseases. What are responsible for the diseases are the microorganisms that are being carried by the vectors. Now let's look at some disease vector, their causative organism, and of course the diseases that are caused, okay, by them. Number one, we have the female Anopheles mosquito. Now this is a vector that carries a microorganism known as plasmodium. So this plasmodium is what is responsible for the disease known as malaria. So vectors are carriers of disease causing microorganism. They carry micro that causes diseases. Just looking for a way to make it easier for you to understand so that you don't make mistake in your examination. Number two, we have tsetse fly. Now this fly is responsible for carrying a microorganism known as the trypanosome and is responsible for a disease called sleeping sickness or trypanosomiasis. Now, number three, we have the house fly. Now, it carry the microorganisms such as Vibrio cholerae and Salmonella typhi. They are responsible for the disease known as cholera and typhoid fever. All right. Now, we have Aedes mosquito. Now, it carries a form of virus that causes yellow fever. We have rats. Okay, they carry a form of virus that causes Lassa fever. And then we have black flies. Okay, they carry a kind of a nematode responsible for the disease known as river blindness. So these are images showing some common vectors in our environment. So you can study more, as many of them as you can get, and know their causative organism and of course the diseases they cause. So at this moment, I'll say we are through with our class for today. So this is a student activity, like a classwork for you. So just make sure you try it. All right, go through the question, answer them. If you don't understand, go back to the video, listen to it one more time or as many times as you can, okay, and ensure that you're able to answer all the questions. And I believe at the end of this you'll be able to tackle any question or 99% of questions on microorganisms around us. So take your time and answer the questions. Okay, this is the answers to the student activity. I won't say we are through with our lecture for today, and so all you need to do now is to make sure you mark yourself, and I hope you have 100%. That is my wish for each and every one of you, that you do well in your exam, you understand biology better. Okay, so don't forget to watch my video, subscribe, introduce this YouTube channel to your friends, view, like I said, drop comments. If you have any comments, any question, just drop it, and as soon as I get them, I'll give you the answer. So I want to thank you for joining me in this class. Thank you for learning. Have a wonderful time.